Hi, my name is Lily Frierson, and today I will be talking about the use of eDNA to study our ocean's microbial diversity. We often hear statistics like 80% of the ocean is unexplored, or it's estimated that 91% of ocean species have yet to be classified. But what do these statistics actually mean? It's hard to say, but they do tell us that there is still an awful lot to explore when it comes to the ocean. I like to think of these statistics as a reminder that the ocean is our final frontier, and there's so much to explore when it comes to our oceans. When we think about ocean discovery, we might think about discovering a new species of shark or a new habitat like hydrothermal vents. But in reality, the greatest space of untapped information comes in the form of microbes. It's estimated that 90% of the ocean's biomass comes in the form of microbes, which is almost impossible to imagine. And in our oceans, these microbes perform a variety of functions. Some microbes are primary producers, like these cyanobacteria. They form the very base of marine food chains. Other microbes are involved in symbioses with animals, like these zooxanthellae, which live on corals and provide them with essential nutrients. Other microbes are involved in nutrient cycles that affect the global scale, like these radiolarians, which are a part of the silica cycle. Others play major impacts in our ecosystems, like these dinoflagellates. When these bloom, specifically the species Carenia brevis, they cause red tide blooms, which can impact large marine mammals and cause major fish kills, and also impact human health. Like all microbes, microbes of the ocean have a rich diversity that we are only beginning to understand. Science is still catching up to these microscopic marvels, and like any microbiome, the ocean microbiome has a lot to teach us. But there's so many, and they're so novel, and it's hard to sample such a big environment, and many we can't even culture. So, how can we keep up with this abundance of diversity? Through the use of eDNA. eDNA is a practice in which genetic material is taken from the environment with soil samples, water samples, air samples. There's different ways to do it, rather than directly coming from an organism itself. So, researchers collect the environmental samples, they extract the DNA with conventional methods like high throughput sequencing or PCR, and then they can use that information to characterize and identify the present organisms. With eDNA, we can sample the ocean in a non invasive way and sample a breadth of organisms. We can gather and track data over time. We can study organisms without culturing, since many of them can't even be cultured. We can find organisms that aren't in a great abundance and identify ones that we don't know by studying their functions through their genetics. We can operate on a vast scale, and with this technique, we can tap into the unknown information held by our oceans. eDNA is expansive and innovative, and it's a new way to study biodiversity in a non invasive, efficient manner. But why is this important? Well, researching these organisms gives us the potential to better understand our ocean's food webs and the fundamental roles played by microbes as primary producers and decomposers. It helps us to better understand how the ocean regulates global supplies of nutrients like carbon and how it regulates our climate. It helps us understand community health and better understand ecological events like bottom up trophic instances. There are also biomedical applications that come with understanding the metabolism of these microbes. Ultimately, with eDNA, we can better understand how our ocean functions and how to keep it healthy. In the wake of climate change, understanding our ocean's health becomes even more critical. As our ocean responds to warming waters, pollution, and acidification, the more we can understand as scientists, the better we can do towards conservation efforts. Knowing the present diversity of organisms, from microscopic microbes to our largest blue whales, as well as these organisms' role in the ocean, is critical to how we can take care of our oceans. The use of eDNA allows us to explore our planet's final frontier, the ocean, and in doing so, we are given endless possibilities for new discoveries. With a focus on microbial life, we can understand the natural sphere in unprecedented ways, paving the way for a healthier planet of the future. The importance of conserving our oceans ends with us, but it starts with microbes. Thank you.